Hello and welcome to Bioinformatics for Infectious Diseases. We hope that in this program, in this session, you will learn a little bit about bioinformatics and how it's used to study and characterize different types of infectious diseases. And I hope that after this webinar, you will be familiar with some of the ways that bioinformatics is used in COVID-19 research, as well as the design and development of novel antiviral drugs and vaccines. And we'll give you a little bit of a perspective on how such data is being used and how you yourself can potentially use genomic data to apply to different types of infectious pathogens. After this webinar is over, you're also welcome to explore our upcoming program called Bioinformatics for Infectious Diseases. It's a training program that will allow you to be effective at using some of the types of tools that we will be discussing today, and hopefully you'll get a good sense of what that will look like. So in this upcoming program, one second, sorry. So today we'll talk about how the emergence and development of bioinformatics in different types of tools for infectious disease research can be used to characterize infection from different types of data perspectives. And we'll also present several use cases for bioinformatics analysis methods that are used in pathogen diagnostics, viral evolutionary studies, antiviral drug discovery, vaccine design, as well as the characterization for emerging drug resistance. And to showcase some of these ideas and some of these types of projects, we'll actually use the T-BioInfo analytical platform, and we'll talk a little bit about several sections specifically designed for such studies that were made available as a user-friendly interface that you can also try. So we'll talk a little bit about what the infectious diseases are, how can we characterize and study them from a genomic perspective, what are some of the tools, methods, and data sets that are available, and then we'll talk about this upcoming program that will allow you to learn how to use these tools effectively. I do want to mention that a lot of the materials that we'll be discussing today have been designed and developed by a team of researchers and a team of bioinformaticians that work at Pine Biotech, but also at the University of Haifa in Israel in the Tauber, excuse me, at the Tauber Bioinformatics Research Center. So these include the lead scientists that have participated in research together with other prominent labs around the world. We will also talk about some of the examples briefly that have come out of those studies. A lot of the methods and tools, as well as the resources that we will discuss in the context of the upcoming Bioinformatics for Infectious Disease Research program, will also be incorporated, and those have been designed by our team of mentors, including Julia Panov, Mohit Mazumdar, and Harpreet Kaur. And so even though I will be speaking, I will be presenting different types of analyses and content that will be presented in that program. And this was a team effort for all of us. So let's start by talking a little bit about what infection is. And so infectious disease is the infection, the result of infection. It's the invasion of an organism's body tissues by disease-causing agents. So it's their multiplication and the reaction of the host tissues to the infectious agents and the toxins that they produce. So an infection disease, also known as a transmissible disease or communicable disease, is an illness resulting from an infection. Infections are caused by infectious agents called pathogens, and they include viruses and related agents such as viroids, bacteria, fungi, parasites, and those parasites could include unicellular organisms like malaria and macroparasites like worms. So non-infectious diseases, as compared to infectious diseases, are also recognized as chronic and can be caused by such factors as environment, nutrition and lifestyle, as well as genetic inheritance. The World Health Organization recognizes four main types of chronic and non-infectious diseases, and those include cancer, cardiovascular diseases, chronic respiratory diseases like asthma, or chronic obstructive pulmonary disease and diabetes. Now, as you will see, a lot of these are actually not so clearly separated. A lot of them have to do with each other. For example, chronic diseases might be a risk for infectious diseases, and infection can cause development of chronic diseases, as we know about cancer, autoimmune diseases, and many others. So in fact, the separation between these two types of diseases is not very clear. There's a lot of overlap, and there's also a lot of different types of cause and effect type of associations between these different conditions. The replication of pathogens in the host leads to major types of interference, and this starts from membrane disruption, as in the case with viruses, and the release of toxins, as in the case of many bacterial pathogens. Many of these interferences induce molecular biological pathways designed to fend off intruders and signal to neighboring cells of the upcoming danger. These pathogens can also modulate the pathways and cause long-term organ damage, such as lung tissue scarring due to the immune hyperactivity. So in fact, the same pathways that were designed to prevent and stop this kind of infection can also lead to major types of effects later on. Now, the hosts can fight infection using their immune system. So here we have a few examples of how that happens. Mammalian hosts react to infection with an innate response, often involving inflammation, followed by an adaptive response. Vaccines are used to train the immune system and to identify pathogens and store the necessary epitopes for antibody production in the long-term immune memory. But due to a variety of factors, such immune responses can actually be insufficient on their own. So two main approaches that have significantly reduced death from infectious diseases in the past century are antibiotics, antiviral drugs, and vaccines. Vaccines have been discovered first as far back as the early 1800s and have become major instruments to train the human immune system in response to various pathogens, reducing worldwide deaths significantly in the 20th century. As a result of water treatment, use of vaccines and antibiotics, death rate from infectious diseases was reduced over tenfold in the 20th century. But the 21st century came around with significant new challenges. Antibiotic-resistant bacteria and the spread of such bacteria even in places like hospitals, as well as significant new challenges from novel pandemic zoonotic viruses that we were not prepared for. And of course, here you can see several examples. This is a brief timeline of novel vaccine design, and you can see that this type of an approach to vaccines has led a lot of people to think that we are done with infectious diseases in the 20th century, and then the 21st century came around, and of course such new challenges like swine flu, bird flu, Ebola virus, and COVID-19 have really shown how unprepared we are for these viruses that are emerging and causing pandemics. Over here, you can see how significantly the reduction of death rate has been as a result of different health epidemiological measures, such as use of chlorine in water in the United States, use of penicillin, vaccines, etc., has really reduced the death rate. And of course, here you can see the opposite picture, what happened in 2020. Here you can see the comparison of the rate of death from COVID-19 to 2017 death rates from other types of conditions. For example, drugs and alcohol, diabetes, lung and tracheal bronchus cancers, and pulmonary disease. So you can see that COVID-19 is actually higher. The death rate from COVID-19 has been going up. And we know that the story is not over, it continues. And so we'll see how bioinformatics can help in some of these challenges. Thankfully, at the same time that the pathogens were studied under the microscope, humans also started developing sequencing technologies that could help decipher the human genome. And as a result, such detailed and rich molecular data has transformed our understanding of infectious diseases and pathogens themselves. In the last 20 years, availability of genomic data has led to an increasing understanding of how cells function at the molecular level and has allowed us to understand major causes of deadly diseases. With the current rise in viral outbreaks and their deadliness, there's a major focus on developing tools and data repositories for a much better understanding of the emergence, prevention, and treatment of infectious diseases. As a result, the major advances in data generation, analysis methods, and the utilization of insights from such studies is making a completely new approach to diagnostics, disease characterization, and the development of novel interventions to such pathogens. So what you can see here is the way that sequencing is used to study pathogens. We know that we can now assemble genomes and accurately compare them between each other to understand how they are interrelated, and also develop genetic markers so that we can understand what are the drivers of virulence and spread, and how they can be helpful in diagnosing and detecting early indications that there's resistance to drugs. And of course, one of the major capabilities of sequencing is that this is digital data. It's easy to store, manage, and analyze. And so as a result, there are many public databases and data sets that allow us to ask very detailed and specific questions about some of these pathogens. Now, as a result of this capability of genomic sequences, we can now understand and study population structure and evolutionary history. We can also identify the specific sequences and very specifically identify the cause of different types of outbreaks. We can have insights into how to make such technology to identify the specific strains early on, and hopefully take early on measures for that. And then also we can use some of the downstream analysis results to inform drug design and vaccine design, as can be shown here. And this is taken from New England Journal of Medicine. So genomics and bioinformatics have contributed immensely to our understanding of infectious diseases, and we can actually apply sequencing data to study disease pathogenesis, or how these pathogens cause disease. We can study mechanisms of interaction between proteins of viral and bacterial pathogens and host pathogens, or their hosts. We can also study the spread of antimicrobial drug resistance and even the host immune response in a very detailed way. So the result of these bioinformatics tools and the sequencing methods has led to the emergence of genomic medicine that has substantially advanced our ability to detect, study, and treat infectious diseases. So one of the major impacts of genomic medicine has been on infectious diseases over the years. So as a result, the ability to perform such bioinformatics analysis, leverage some of these tools, and access these large data sets is critical for all those of us that are participating in such research. Omics data is now regularly used to study viral genomes, DNA variation, host gene expression, protein abundance, and composition properties of microbial communities. And all of these are very different types of data that all together form a comprehensive, holistic picture of how these pathogens interact with the host, cause disease, but also live in communities that we need to learn how to study and understand. So what we'll do today is we'll take a look at several examples of how such data could be analyzed and what type of information such data can provide us with. So the role of bioinformatics in general in the context of infectious diseases will allow us to study such data to extract important information. And so what kind of information can we actually extract? We can talk about the association of viral genomics and evolution, placing that in the context of time and geolocation. So we can ask questions about where did a specific pathogen originate? How does it spread in the population? What are some of the different phenotypes that it produces in the population? We can talk about immune response and really define how immune response is changing in the course of disease progression, and ask questions about what are those specific phenotypes caused by which genes, and maybe identify molecular pathways that we can interfere with, leading us to novel drug design and combinations of drugs that could be effective at suppressing disease manifestation, as well as addressing the specific pathogen. So how do we interfere with the pathogen production, colonization, and the production of toxins? And also, it allows us to understand better the conditions in which these pathogens thrive and how to interfere. So for example, a large component of the immune response is that initial microbiome composition. So we can ask questions about how does that composition change? What are some of the factors that affect the changes, and how we can be more informed about the approaches to intervene and to preserve a healthy component of this immune system or the immune response in the microbiome. So one major way that bioinformatics is actually changing our understanding of the microbial world is the ability to ask very basic questions about individual organisms, their functionality, environment, and the communities that they form. And in fact, this new sequencing technology and the methods have allowed us to not only look at individual pathogens, but also study these communities as a whole. And so a little bit of what we will touch upon is the diversity of viruses and bacteria, as well as their interaction with the microbiome and the virome that could be studied as a single community instead of just individual pathogens that we identify. So what is the virome? The virome is one part of the microbiome, which constitutes all of the organisms that inhabit us, including bacterial and archaeal organisms, as well as the microbiome and other members of the meiofauna, such as protists and metazoans. Studies of the virome are actually in their infancy because it has only recently become possible to see and study the virome in large sequence data sets using bioinformatic tools that can detect relationships between viruses despite their extreme nucleotide sequence diversity and dynamics, because they evolve and rapidly change. The size of the mammalian virome is actually not known, and we think that there is a tremendous diversity, and it still has to be studied further to really understand how diverse it is. And it is continually updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. So it's a fairly complex picture or a puzzle that has to be assembled together before we can ask specific questions. But what already is clear from the ability to analyze sequences are some questions about the very basics or even life itself. So biologists have categorized life into three large domains: bacteria, archaea, and eukarya. We know that viruses were originally not even considered in this tree of life, but these novel abilities to compare sequences and identify genes that are responsible for a specific type of functionality have really opened our research to understand viruses as potentially participants in the dynamics and the diversity of life itself. And we can think of them as agents of change because they have to be able to replicate themselves using their host cell machinery. And as a result, there's a lot of exchange of genetic material that happens between viruses and their hosts that include bacteria, as well as eukarya and archaea. And so as a result, we see this exchange of genetic material between these different hosts, and we now can think of viruses also as maybe potentially a part of this tree of life in general. So before we continue, and we'll talk specifically about some of the tools and methods that could be used to analyze such data, let me pause here for a second and ask, first of all, if you have any questions before we continue, but also let me ask you a couple of questions as well. So I know that there's a lot of people here. Some of them are online on YouTube and some of you are online on Zoom. So if you're on Zoom, I would like to ask if you could put down in the chat box, where are you from and what institution are you in, working in, studying in? So really, where are you located geographically, and what institution are you in? So if you could use the chat box and just let us know, that would be great, just so that we have some sense of where you are right now. All right, thank you, Dr. Hunduma. I see that you're from Ethiopia. And please don't forget to mention what institution you're a part of. Okay, Adama Sciences and Technology University. And I see Southeastern Louisiana University. Thank you, Caitlin. Sheffield University, the Neuroscience Department. Thank you. Kenya, KEMRI. Thank you, Meshach. Nairobi, Kenya. Great. All right, well, Applied Biology Department, thank you. Osmania University, India, Kenya Medical Research Institute, African University of Abomey Calavi, Benin. All right, excellent. So thank you for sharing this information. This is going to be useful for us as we plan these workshops for the future. So again, for anyone that is joining us, we welcome you from all of these locations, and it's great to have you with us today. So what we'll, okay, thank you. And we have somebody also here from Lagos, Nigeria. So it would be great to continue and stay in touch and to really benefit from some of these future webinars and some of the resources that we provide. What we ask you to do, if you would like to continue and participate in some of these in the future, is join our social media groups so that we can stay in touch. So we have a group on LinkedIn where we discuss a lot of the emerging topics, but also have posts about different research opportunities as well as career opportunities. So if you are interested in bioinformatics and omics data, please join our community on LinkedIn as well as on Facebook, and we can continue and share some of these updates there. Okay, so let's go on. We'll talk next about next generation sequencing, and really how can next generation sequencing data help us ask some of these questions in relation to infectious diseases. So one of the major challenges in omics data is, of course, the ability to process the data and extract meaningful information that we can use to ask very specific questions using analytical tools. So this challenge is to process, visualize, and organize data so that these questions could be answered. In metagenomic and viral sequencing, there are three types of sequencing approaches that we're going to be talking about today. There's what's called targeted sequencing, where we can amplify a particular pathogen or a type of microorganisms, a specific taxonomic classification. So we can talk about different types of primers that allow us to sequence a very specific type of a genome. We can also talk about amplicon sequencing, and amplicon sequencing means that we amplify a particular region, and this could mean that we combine specific library preparation techniques as well as PCR amplification to have enough material to study diversity, for example. And then, of course, we can sequence everything, and sequencing everything can yield all DNA, so all of the genomic material, or all RNA material. And so when we talk about gene expression data, the response of the host to infection, we can study that using RNA sequencing. So we'll talk about some of these different methods. Importantly, a lot of this type of data is actually organized and made available to the general public. So here you can see an example of a couple of different places where you can find such data. The Joint Genome Institute, as well as the NCBI, provides us a very good view of how much data has been available. And there is a lot of rapid growth and accelerated increase in the number of available sequences. Now, many of these data sets are structured. In other words, they are already assembled genomes, and these assembled genomes could be viewed by a particular name. So you can go on this new project, for example, called Ncbi Virus, and you can actually find specific assembled genomes or specific sections, genes and proteins of those genomes. And there's a lot of what's called bioprojects or SRA data sets that you can actually study for novel types of tools that allow you to compare different components and sequences and gene expression data. And so it really provides us with access to a very diverse set of data sets that we can use to ask specific types of questions. The data that we have access to can also provide us with rapid identification of novel sequences. So you can see how such viral outbreaks measures can be studied using next generation sequencing. For example, we can take data specifically from clinical specimens and identify either a particular viral sequence and then study that sequence and how it's different from other sequences that we already know about, or we can actually look at metagenomic sequencing and analyze all of the data that is available there, potentially looking at multiple types of viruses that are available, multiple types of pathogens that are available, and then study variation and composition of these communities within each sample. Here you can also see some of the additional ways that we can apply next generation sequencing data analysis tools to identify, detect resistance to antimicrobial, antibiotic resistance, analyze changes in the microbiome community, and of course study the host gene expression or transcriptomic data. Now, I mentioned already that the ability to analyze such data is complicated because this data is unstructured. So essentially it consists of short reads. You can see here millions and billions, in fact, of reads that could be extracted from a single sample. And one of the challenges to make sense of what does this data actually mean. And to do that, we can assemble these short reads and analyze those reads, linking them to functional components. So another important repository here is the repository of reference sequences, sequences of either specific organisms that we already have studied and understand their functionality. As for example, in here, you can see that one of the first steps that had to happen when this outbreak of this novel coronavirus happened is that we had to have a good reference of this genome so that we can map the reads onto the particular genes in the genome and understand their function to study how does this virus actually function inside the host. So once we have that sequence and we can map the reads that come from novel sequencing samples onto the reference genome, we can in fact start asking questions. So what kinds of questions can we ask? One question that we ask is sequence variation by aligning new sequences that we have to a reference. And we can ask this about the virus, we can ask this about the bacteria, we can ask about any genome that we have by just mapping them onto a reference or by mapping them together to identify similarities between different components that we have identified. Now, to ask a question about how they are related, we can also conduct evolutionary analysis by looking at relationships between these sequences and organizing them into a phylogeny, or how they are interrelated by coming from some common origin. We can also then ask questions about how the differences between these genomes or genomic elements like genes are functionally leading to changes. So here you can see a mapping of mutations on one of the proteins produced by a virus. And the question that we can ask, well, how does that actually affect the interaction between this protein and a corresponding host protein that could mean a change in the way the disease happens or the way that the transmission happens. And then we can also ask questions about the host response, and really that is a different approach. Here we are more looking at the expression at the levels of genes, how the host response, the expression of genes varies between different types of infections and at different stages of infection, causing us to understand how the immune response varies between different cells, of course, in different tissues. Okay, so one of the things that I would like to introduce now is some of these approaches on the T-BioInfo platform, where we have developed easy-to-use tools that you can try and use for analysis of phylogenetic relationships by identifying and mapping relationships between genomic elements that are different between each other, how you can study gene expression by analyzing transcriptomic data, alignment of sequences to each other to detect prominent mutations and variations that we can link to their functional changes, how such data could be taken to the structure to understand the relationship between sequence and structure, and how variant calling and identification of particular markers of these genomic changes could help us understand why such structural changes are happening. So let's first talk about the alignment, the process of alignment. So multiple sequence alignment is a typical approach to identify changes based on nucleotide variation, as well as based on amino acid variation. So to analyze such data, we need to understand the differences in multiple genomes and to compare those genomes based on areas that are matching in those sequences. And importantly, to understand the functional implications of such changes, we also need to understand the relationship between nucleotide sequence and amino acid sequence. So let me give you an example. I'm going to go to the server platform. So this is the link, and perhaps some of you that are not joining us for the first time might already be aware with this platform. The platform was designed to incorporate all of these different methods of analysis into a single interface and make it easy to use for even someone that is not that does not have a technical background, an engineering computer science background. So one of the ways that you can try out some of these methods before you actually use this platform for research is by requesting a free demo account. And the way you can do that is you can go into this Access tab right here and fill out this form, and you will actually get a free demo account that you can try out some of the things that we are going to be doing now. So I'm going to show you a brief example of what this multiple sequence alignment really looks like, so that we can then try and apply it to an example data set. So here I'm going to go to this virus quasi-species section and going to go into this first demo for multiple sequence alignment. This whole area is actually designed for multiple use cases. You can see that you can identify point mutations in SRA files or FASTQ files, where we are expecting to find a whole diverse population of, let's say, viruses. You can also identify function of defective viral genomes that might have large, long sequences deleted from the full genome. And you can also study relationships between the sequences that have been assembled into haplotypes, or if you have consensus genomes, you can also understand the diversity of these consensus genomes. So a very basic pipeline that could be made here is based on this data set of multiple types of coronaviruses, which include the novel SARS-CoV-2 genome, the bat genome of a similar virus, and then also SARS-1 genomes. And so the question might be, how are they different and what do those differences mean in terms of their amino acid substitutions? So here I'm going to just click on OK. And you can see that I have a section here for multiple sequence alignment. I can press on multiple sequence alignment that allows me to understand specific similar and different sections on the genome. Converting those nucleotide sequences into amino acid sequences, which will give us insight into how different physical and chemical properties of those regions are changed. And I can then ask question, well, how likely is it for such a change to occur? And also, what kind of implications for this substitution will I expect to find on the functional level? So here I'm going to click OK. Importantly, we know that the evolution or the accumulation of mutations does not happen at the same rate in different types of codons. And so I can also include different variation to how I look at the specific codon positions to analyze this data. And then I want to build a phylogenetic tree that essentially uses multiple sequence alignment to determine relationships between sequences and assign time based on different models. And you can see here a number of different models that I could use, or I could just combine all of these phylogeny methods together. And essentially what I will get as a result is the relationship between ancestors and the present day species based on some assumption about how that rate of mutation can lead to the changes that I observe in the samples that I have in my data. Those trees could be analyzed, and I can combine all of the trees that I get as a result, because each different method will produce a different result, and then select some of the most representative trees that I think are more linked to the actual data that I have based on comparison between multiple trees. So clicking on End, here will give us the results. I can open this folder and take a look at some of these results and really start asking questions about what does this mean. So let's first of all take a look at some of these results. I have here a ape tree, which is one of the methods, and then here I have the beast birth death process tree. So we can take a look at what do they look like and how can this give us insight into what these different types of sequences, what type of information do they contain, and how are they organized into this relationship. So here you can see that this phylogenetic tree, a rooted phylogenetic tree, contains 1, 2, 3, and 4 defined groups. And one of the questions that I might ask is, how does the data that I have about these sequences can help me understand what SARS-CoV-2, for example, what is the closest relative for SARS-CoV-2 out of all of the sequences that I have uploaded into this analysis. So beyond just having the ability to look at a phylogenetic tree or look at the sequence variation, or maybe ask the question, what is its relationship and how does this affect the amino acid and the protein structure? Ultimately, I would like to link all of this information together. So to do this, we have actually created a visualization tool to help ask those specific questions. So the same tree that you are seeing right here, the same sequences that we see right there, here they are in a more visual and interactive format. You can see in this tool, which we call Sequence Navigator or SeqNav, you can see the sequences of the genomes, and those sequences are organized by groups. Here you can see MERS, SARS, COVID-19 or SARS-CoV-2, and beta coronaviruses or other coronaviruses that are not SARS or MERS. And the cells are bins. Each bin represents 500 nucleotides. We can make it also into 250. So let's take a look at 500 first. Well, okay, so they're loaded. So here we can see that each one of these bins represents a bin of 250 nucleotides. We have the reference genome that it is mapped to, and the difference is color-coded from gray all the way to this darker red. So right away, what we can see here are some of the groups are different in terms of how similar they are to the reference genome. So based on what you see, and you can, by the way, navigate to this link yourself and try it out for yourself, which group do you think is the most dissimilar to this reference genome? Okay, you can type your answers into the chat box. Which group of sequences is the least similar to the reference genome? Again, gray is the most similar, red is the most dissimilar, the most distant, based on the number of changes that are observed in that bin. Each bin is about 250 nucleotides. So which group? You can see that there are groups MERS, SARS, COVID-19 or SARS-CoV-2, and beta coronaviruses. So which group do you think is the least similar to this reference genome? Okay, so Caitlin is saying beta coronaviruses and Mishak is saying COVID. Kate, okay, is also saying beta coronaviruses. So let's see what might be some indications for this. As I mentioned, gray is similar, right? So if I look at this whole group, it is very similar because in fact, I don't have any variation here between gray, yellow, and red. And so as a result, I know that this whole group, which is COVID, is the most similar to the reference genome. And the reason for this is, of course, because the reference genome is SARS-CoV-2, and so it is exactly the same reference genome. Now, if I want to look at other types of comparisons, I can, for example, take a look at the COVID-19 human genomic sequences and ask the questions, how do these SARS-CoV-2 genomes differ between themselves? But in the context of these comparisons, this group, SARS-CoV-2, is the most similar to the reference genome. So the question is, which type of a virus or groups of viruses are different from the SARS-CoV-2? And the answer is that indeed, like you mentioned, either this group, which is MERS, or this group, like beta coronaviruses. So beta coronaviruses contain a lot of red in specific regions, right, including this region right here and this region right here. So let's now talk about what might this mean. So I have here SARS, which is SARS-CoV-2, which is very similar to SARS-1, and quite dissimilar with MERS and beta coronaviruses. So one question I can ask, well, how are they related to each other? In other words, which one is closer and which one is farther away? And so the answer to that is this phylogenetic tree. The phylogenetic tree tells me which groups are closely related to each other and which ones are farther away from each other. So here you can see that COVID-19 and SARS-1 are closely related, and they are closer related to MERS than they are to the beta coronaviruses. So even though just looking at the sequence alignment, I don't really know which ones they're closer to because there's so much variation. If I look at the phylogenetic tree, which gives a model for how one type of a sequence could originate from another one, I can start organizing these into relationships. And these relationships tell me that in fact, they do differ in terms of how closely related they are to each other. Now, the other thing that I can ask is, how do the groups within each defined group or each clade relate to each other as well. And I can see that they all do have differences as well. And these differences are actually related to time. And so I can see that beta coronaviruses have the greatest diversity in terms of time, just because I see that this axis, which corresponds to evolutionary time, shows me a greater divergence between the actual sequences, and also in this group right here, which is COVID-19, that diversity almost does not exist, which is very logical, of course, because COVID-19 has been around just for such a brief time, and this time incorporates such viruses that have been around for years and years. And so the diversity of the samples that I have in the different groups actually represents very different physical time or real time, and therefore this evolutionary time differs between diversity measurements in each one of these groups. The other question that I might ask is how the differences that I observe at specific positions on the genomes could be interpreted by looking at what proteins they encode for. So here I have this reference genome that also shows the different proteins, and these proteins could be, or these genes, and here I can look at these genes and what actually they do. So you can see that one of these additional areas that have a lot of dissimilarity is the surface glycoprotein, or the spike S glycoprotein. So here I have this spike glycoprotein. We know that it is responsible for interaction with the host protein for cell entry. I can actually see this 3D structure of this protein and read about what it is. And I can actually go to this link, which will take me to the Ncbi, where I can find more information about this protein and that full sequence. So this is an important component as well. Not only am I asking the question, how do these different sequences related? What areas are mostly related or most dissimilar, but also I can ask the question of what might be the functional result from changes in that protein. And so as you go deeper into these comparisons, more and more questions could be answered by studying these sequences. Inside each sequence, remember that there's 250 nucleotides. And here, if you hover over, you can see how many of those are actually different from the reference. So you can see that this very red one has 220 changes from the reference. This one has 57. And of course, a gray one can have 0 or a very small number like 3. So this tool allows us to interpret some of the results from such sequence analysis, from such multiple sequence alignment and phylogeny, to start understanding what might be some differences in terms of where these genomes originated from and how these genomes could be different in the way that they interact with the host. And here, since we see so many changes in the specific spike glycoprotein, we can start asking additional questions about what specific changes are there and how can they explain the differences between what this virus does and how does it actually cause disease. So this is an important kind of a tool in the toolkit that we have to compare assembled sequences by aligning them to each other and understanding what types of mutations or changes we can observe in the sequences that we have in our data. Now, importantly, not all of the changes are represented equally in the data that we have. If we have full FASTQ files, and these FASTQ files actually represent specific sequences from reads, short reads, we can see that some of the changes in those reads could be of high confidence. For example, if I have a read that I can detect in multiple reads and all of them agree, they give me the same result, and I know that it is a change from the reference, I will be confident in that change. But in some cases, I have a lot of variation. So even though I see a change, that change is very different inside different reads. If it is different inside different reads, I really cannot call this a variant. And so the variation between the different reads in the sequences of the reference genome have different weights and different types of information that they can give me to understand how those mutations are accumulating in the population of viruses that I'm studying. Now, for this purpose, it is important to call variants and distinguish between the different types of variation that I have in the data. So here we have an example. You can see that this is coverage and identification of these specific types of positions where I have high confidence is also carried along with positions that I have low confidence. And sometimes the proportion between them changes as well. So this is an additional piece of information because the reality is this typically represents a diverse population of sequences that accumulate as the virus replicates inside the cell. So if I'm looking at this situation, I have a particular original virus coming into the cell or coming into the host. It replicates and has to evade multiple pressures associated with the immune response and competition between different progenies of this individual virus. And as a result, a lot of mutations occur, and I can actually study the variation of this population using some of these mutations that accumulate in the individual genomes that I can sample. So if I take a sample, one single sample, let's say from a patient, and take another sample from a different patient, I can actually ask the question, not only how the genome in each one of those of a specific pathogen is different, but I can also see what diversity levels am I observing. And oftentimes these diversity levels are associated with virulence and the danger that they can carry for the immune system. Because as the virus mutates, it is the only strategy essentially that it has to evade the detection and suppression of what it does inside the cell. And so as it accumulates these mutations, some of those mutations can become more significant. In other words, the virus with that particular mutation becomes more abundant. And in some cases, some other mutations can become less significant. So they can really represent some kind of a dying out population. Now, again, to piece all of that information together, I can also organize the relationship between these different types of viruses into one single tree, and then I can study these trees to understand populations. And this type of an analysis, phylogenetic analysis, can be very powerful for me to start associating with whatever I observe at the phenotypic level. So a good example of that is, of course, this visualization that probably most of us are already familiar with, that allows us to link how time, diversity in the viral population, and some phenotypic information about the location or maybe the number of people that were infected in a particular region could all be linked together. Now, importantly, we see that these mutations do not accumulate equally in each viral genome or in each viral gene, sorry. So the prominent mutations, the mutations that start emerging in a particular region on a particular time frame, are important for us to study further. Now, what kind of information can we extract from a carefully selected number of sequences when we start looking at the in-depth meaning of these mutations? Let's take an example. The example that we'll look at is the example of this specific comparison between changes in COVID-19 genome as compared to some of the SARS-1 genomes and the bat coronavirus genome, to try to ask the question of what might be the probable origin and how might the virus originate from a similar species like a bat or a pangolin as compared to the coronavirus that we observe in the human. And so to do that, First of all, an important part of such a question is finding the right data. And typically, the way that we can select for the right type of data to ask such questions is to first analyze large quantities of data to study variation overall, and then to reduce the number of genomes to a very particular set of genomes that we have high confidence in. So these could be consensus genomes. And based on that consensus genome, we can start asking very specific questions about individual positions on the genome that show some major change that we think have a functional implication. So for example, if we start from an analysis overall, here you can see how analysis between many genomes could be converted into a comparison of overall similarity across the genomic positions and identify very specific regions that show high difference. Now, that high difference could now be interpreted by asking very specific questions about the positions that are different and the distance between the positions. What do I mean? So here we have an overall kind of an overview of how we can take this study further. For example, if I know of a particular protein, just like we saw the spike glycoprotein, has a lot of variation. Within that sequence of that protein, let's say the spike glycoprotein is about 1,500 nucleotides long, 1,500 amino acids long. So we can start asking the questions of how can we identify the functional significance of those changes. So here we see amino acid sequences that we can analyze in terms of sequence identity. How are they similar? Which context are they placed in? And also what types of features can we identify in those genomic sequences? We can also look at the 3D structure and think about how can we position the changes that we observe in the sequence on this folded structure. So which ones are outside, which ones are inside, which ones are interacting together, which ones are more distant from each other. And obviously, we can also generate high throughput data that shows us the response of the host that could be different because specific positions have changed. So here is an analysis of such data for this spike glycoprotein. And you can see that this is the percentage similarity on the reference genome. And you can see that the major changes occur in the spike protein. So these major changes across groups of different sequences reveal the changes that we can start looking into deeper. And we can separate the spike glycoprotein into different functional domains. And these functional domains are corresponding to the different types of activity that this protein actually assists with. So if we look at this receptor binding domain, the specific position of mutations that we can see, they accumulate significantly in the receptor binding domain. And we can link that to our understanding of the overall interaction between the viral proteins and the host proteins. For example, we know that it interfaces with the human protein that sits on the membrane, right? It is a protein that is responsible for cell entry. And so we can see how the specific protein of the virus, the spike glycoprotein, interacts with the ACE2, which is hosted inside this protein called B0AT1. So the interaction between the virus and the human cell is first interfacing in the receptor binding domain and just one part of this ACE2 B0AT1 complex. Now, as we look into this receptor binding domain, we can see that it is only about 200 nucleotides long. And this 200 nucleotide sequence could be studied in much greater detail to reveal how the particular changes that we observe in the genome are responsible for adaptation. because we know that the complex that we observe here and the specific receptor that is ACE2 in humans is also different between different species. So by analyzing the genomic sequences of the pathogen and analyzing the genomic sequences of the host, we can start answering the question of how a particular interaction between different hosts can help us trace the evolution of the virus as it came from other species and eventually started infecting the humans. Now, answering that question conclusively can also help us identify the specific positions in the interaction between these two proteins that could be intervened in or that could be trained for the human immune system to be able to recognize the specific binding positions and the specific variation that is typical in these types of infections that we can start designing vaccines and drugs. Now let's take a look at another example. Here's an example of Ebola virus, and the Ebola virus is also very diverse and has occurred in several different pandemics that were observed in the West Africa region. Now, in that West Africa region, we can take samples, and by the way, this was one of the 2014-2016 outbreaks, was one of the first major efforts in near real-time sequencing that produced thousands and thousands of samples that we can analyze for diversity. Now, looking at that diversity, we can identify the most prominent changes in the viral sequences that were sampled and identify which proteins are they accumulated in. So for example, in this study, you can see how, again, we have multiple reads from many, many samples, from population samples. Those reads were aligned together and to the reference genome. As a result of the alignment, we identify specific positions, and those positions can reveal important proteins where such mutations can play a functional role that could be linked to virulence, transmission, and ultimately inform vaccine design. Importantly, we have today an antiviral drug and a vaccine for the Ebola virus as a result of bioinformatics and as a result of such analyses. So having access to data and access to tools that could be used can help us identify specific changes and ask the question of their functional significance, as well as what type of information could they reveal that is important for drug and vaccine design. But so far, we've talked a lot about viruses. What about other types of pathogens, specifically bacterial pathogens like tuberculosis and such pathogens like malaria, which are actually also developing drug resistance? So for those types of bacterial infections and these types of situations, one of the similar approaches can actually help us reveal what genomic variation is responsible for single antibiotic resistant or multiple antibiotic resistant types of pathogens. So here you can see specific mutations that are associated with different types of drugs where resistance in tuberculosis is developed. Now, that is not done by looking at just individual positions and multiple sequence alignment. If we return to this platform interface, we can see that some additional important sections are available here to ask such questions of genomic data. So for example, here we have genome-wide association studies that essentially help us ask the question, if I have a defined phenotype, and that phenotype is resistance to a particular drug. Let's say I know that a drug is being administered in this region, in this hospital for this patient, and some of it is ineffective. How can I ask the question, what is the association between the specific variation in the genome of that pathogen And what is the association with the phenotype, right? So if the phenotype, sorry, I'm trying to mute someone. If the phenotype, sorry, guys. Okay, there we go. So if I can find such an association, I can actually identify which mutation is responsible for accumulation of drug resistance. Similarly, I can have a more of an approach of multi-omics integration where I have an ability to ask the question, how is a mutation specifically responsible for the interaction with the host? So if I have the host data and I have the genomic data from my viral or bacterial pathogen, I can start looking for associations in such data sets that can reveal interaction between different proteins and signaling pathways and help me identify how to intervene in those interactions. Now, importantly, the examples that I'm showing you are just to get you thinking about what types of analysis, what types of data, and what types of questions you might have about a project that is interesting to you. So these examples are based on some publications that what we did is we took the data from those publications, we looked at the methods that they used, and then we combined those together to produce tutorials that could be used as a starting point for a research project. Now, if you're a student or you're just learning about bioinformatics, what you might want to do is you might want to do some similar analysis and develop a novel and independent project. And such a novel and independent project can really give you a sense of how such tools can be used for this type of data. Now, as I mentioned, such data is widely available. One example that we can briefly look at is on Ncbi Virus, where a lot of this data is hosted and is freely available. So I'm just going to go to Ncbi Virus first, and then I'll also show a couple of examples from the Ncbi repository where you can look at data sets for other types of pathogens. So in Ncbi Virus, you can start by searching by sequence, or you can look by virus. So if all of you try this tool out, what you can easily do here is enter, for example, Ebola virus. You can just wait a couple of seconds. You can see that the Ebola virus appears right here, and I can access over 3,000 sequences from Ebola virus. And you can see that they come from different locations. They come from different sources at different time frames. So I can really start designing my questions around the data sets that are available right here. Let's go back, and instead of Ebola virus, let's look for coronavirus. So here you can see the whole coronaviridae. And the coronaviridae also comes from different hosts, so animal hosts as well as human hosts. And I can also narrow this for different locations, geographic region, isolation source, and dates. So again, the same type of analysis that we went through today, comparing different sequences and asking what might those sequence analysis results mean functionally, you can also conduct just by taking data from here. Another great repository is for all other types of data, where you can also find host response to coronavirus infection. You can find malaria sequences, tuberculosis sequences, Ebola virus, and other pathogenic sequences, as well as the host response. And you can really start looking at both the diversity of these viral populations, the microbiome that they best develop in or worst develop in, and also the response to different treatments. So here, if you look for coronavirus you will see that there's, of course, a lot of different types of data that you can find, including gene expression data, over 3,000 gene expression omnibus data sets. You can see 351 bioprojects, which in all of these projects together have over 100,000 biosamples. So you really have a lot of access to data sets right here, and over 100,000 SRA, which is the sequence read archive or the raw FASTQ file. So really a tremendous diversity of data sets. If you just look through some of the titles of these bioprojects, you can find what type of data sets are available and think, how can you ask a specific question of these data sets? For example, evaluating the microbial context of SARS-CoV-2 in patients and the hospital-built environment, or super spreading event preceding a large meat processing plant related SARS coronavirus 2 outbreak in Germany. Right? So really fascinating data sets. Many of these data sets have a publication associated with it, and some of them don't because they're very new. So you can see that this was submitted on the 3rd of November. So just recently, there's not even a publication associated with this data set. And you can easily access over 1,000 samples here to start asking questions. Well, what is really going on? And how can I ask this question based on the data that I have available? So I think that this represents a huge opportunity to design and develop novel types of projects that you can start using some of the tools and the data sets that we have discussed today. Now, I also want to tell you a little bit about the training that our company provides to help you get started using some of the tools and the methods that we discussed. We run a series of OmicsLogic training programs that are intended to provide bioinformatics guidance for independent research using bioinformatics with assistance from mentors and peer example. And our goal is to develop a community that has data-driven research interest and an appreciation of citizen science. And we want to make sure that students, clinicians, faculty of all backgrounds are able to apply bioinformatics effectively to these kinds of questions. And so thinking about the interest and the available resources for infectious diseases, we've designed this novel program called Bioinformatics for Infectious Diseases. This program will help you understand pathogens and the diseases that they cause by analyzing data, and learn about the different analysis approaches that can help detect viral genomes in next generation sequencing data, to ask questions about how these sequences are different and apply those types of analyses to different types of functional and important projects like vaccine design, antiviral drug design, antibiotic resistance, epidemiology, and others. What we provide in the context of this project, of this program, are curated projects and data sets, just like what we discussed today very briefly. In this program, we will go through in much greater detail. And so we'll talk about different types of viruses like coronaviruses, and also different types of bacterial pathogens as well. The programs are really combined out of several different elements. They include online sessions like today. We try to make them interactive, and they're conducted by multiple guides that we have in our program. They also have access to self-guided study materials that you can access independently, and some practical assignments. So let me briefly show you what those look like. So the platform that we already talked about is one of these resources. This resource allows you to analyze publicly available data sets easily with a user interface. We also have a number of projects that you can use to get started with your research project or analyze data from other types of data sets. And we have coding exercises, and I'll just briefly show you one of these coding exercises that is very basic, but introduces you into coding practices using R and Python to analyze genomic sequences. So here you can see a breakdown of the code. You can then learn about more advanced variation of the analysis, including pairwise analysis and multiple sequence alignment. And importantly, you get these assignments where you have to modify the code and run it to produce a result. Now, all of this interaction with the coding assignments, with running individual pipelines, or even completing some of the lessons and projects that you will see right here, all of this becomes available under your individual profile. And so you get to build up a profile of your own to demonstrate what types of skills you have achieved by participating in the program, and also develop an independent project. And we have a number of examples that you can also look on our website to see what students have been able to develop as a result of participating in a project like this, in a program like this. So to summarize, the program will start on November 9th. It is called Bioinformatics for Infectious Diseases, and it is a training program, a bioinformatics training program that will help you explore relevant tools, methods, and data repositories, learn from curated case studies and peer-reviewed publications, test your understanding by analyzing sample data, master key elements of analysis and visualization, as well as design and develop an independent research project. So these are some of the key topics that we will cover in this program, and this program will start on November 9th. Now, importantly, we have a place where you can find all of these details called Bioinformatics for Infectious Diseases, and I'm just going to paste the link in the chat as well. On here, you will find the details, including a brief overview of the program, the schedule of the program. So we will have these different sessions together. In each one, you will find a description of what topic will be covered, what is included in the program registration, some of the comments, some of the access to some of the resources that you will have, some of the projects, and some of the tools that we mentioned. And of course, these sessions are just one example of the types of projects or the programs that we run. So you're welcome to explore some of the additional programs that we have available here as well. Okay, so thank you again for joining today for this brief presentation. I am happy to answer any questions now that you might have about what we talked about today, including the program and including some of the topics that we discussed and the project examples that we discussed. So if you have any question, you're welcome to write it in the chat and I will answer those.